Okay, thank you. Good morning, good afternoon, and good evening, everybody. I'm Irawan. Currently, I work for the Ministry of Environment and Forestry Indonesia as a Deputy Director for the Greenhouse Gas Inventory. Previously, I am a researcher at University of Auckland, conducting my PhD with the topic of prioritization of marine conservation in the coral triangle. So it's about the biodiversity, using biodiversity criteria to identify areas with the most conservation value. And today we have a biodiversity Friday. I think it's really nice to have a biodiversity Friday, and you can talk about biodiversity in general term or in the specific term. And we are really happy today that we have two distinguished speakers from different regions of the world. One is Dr. Dinusha Jayathilake, and one is Dr. Qianshuo Zhao from Ocean University of China. And let me introduce a little bit about the background of our two speakers today. Dr. Dinusha Jayathilake will talk about her studies on identifying the distribution of marine biomes on global scale, especially focusing on seagrass and kelp communities. And now, Dr. Dinusha, she is a lecturer in the Eastern University of Technology in Napier, New Zealand. It's a nice place to be a lecturer. And then and conducting a research program on the freshwater ecology on Hawke's Bay region in New Zealand. Dr. Qianshuo Zhao will present his research on mapping a marine ecosystem and biodiversity-rich spot in global scale and in the West Pacific region. He is a lecturer or a faculty member of the Ocean University of China in Qingdao, and amazingly, he won the Elsevier Atlas Award for producing the most comprehensive and entirely data-driven prioritization of MPA for the world. Yeah, I think this without further ado, I give the floor to Dr. Dinusha Jayathilake, because ladies always first. So Dinusha, you have the floors. Time is yours.
Thank you, Rawan. I'm going to share my screen. Can you see my screen now?
Yeah.
Okay, so my research is using marine biome maps to expand the marine reserve network. So before we go to talk about this research, I would like to give a introduction about myself. Ayubowan and Kia ora. So I'm a Sri Lankan and live in New Zealand. And currently I'm working for EIT as an environmental lecturer. I did my PhD in 2020, completed my PhD in 2020 from the University of Auckland, and my PhD title is Mapping Marine Biomes of the World. So today I'm going to focus on the fourth chapter of my PhD thesis, is how these marine biomes can use to demarcate or delineate the marine protected areas. So before we talk about this marine protection, I would like to talk about what is the biome. The biomes are large biogenic areas characterized by the same plant life form. So if you look at this left-hand side, you may be familiar with this terrestrial biomes. Like they have three-dimensional structure, permanent and temporal spatial distribution, feeding, breeding, and nesting habitat for associate fauna. These biomes have unique plant species as well as unique associate faunal communities. So the second picture from the left-hand side shows a savanna, and the topmost picture, the map shows the distribution of savanna. And the picture from the left-hand side shows a tropical rainforest biome. And bottommost one shows the global distribution of these tropical biomes. So this is the concept. It has large biogenic area, and you can see this. The middle picture shows they are totally different, but they are terrestrial biomes. And so my PhD, what I wanted to introduce this concept to the marine domain and to see whether we can identify the similar pattern or the similar organization within the marine domain. For that, I use 4 marine biomes: seagrass, kelps, mangroves, and Zooxanthellate corals. Even the Zooxanthellate corals are not plant-based community or the habitat. I chose it because it follows all the other characteristic features like 3-dimensional structure, permanent and temporal distribution, provide feeding, breeding, and nesting habitats for the associate faunal communities. And even like you can use salt marshes as a biome, kind of like an adjacent biome or the marginal biome, but those salt marshes are more terrestrial than coastal. So I used only these 4 biomes. So the next question is there any use of these biomes? And do we need to study or talk more about these biomes? So simply, there are like lots of socioeconomic uses of these biomes. For example, as a human food, we can eat fruits, seeds, seedlings, leaves, or many parts of the mangrove species. And it's very like highly valued as tourism and aquaculture. Many countries cultivate shrimps, do the farming of shrimps, prawns, and kelp for the fertilizer or the for other food commodities, medicine, fisheries, housing materials, and even the interior decorative items and commercial products like as tannins, soap, and cellulose acetate. So those things, there are like lots of economic uses. And the next question is, do they have any ecological or biological significances? The simple answer is yes. So here I highlighted few ecological and biological significances according to the Archie Target 11, because I completed my PhD in 2020, and that is the cutoff year for the achievement of Archie targets. So here I highlighted those targets parallel to my study. These biomes have unique, rare, and endemic species, population, or communities. For example, seagrasses, there are like a few endemic species, and all the biomes have endemic and rare species, and they have populations and communities, and are essential for associate faunal population to survive and grow. That's true. And contain species, population, or communities with high natural biological productivities. All these are plants or the photosynthetic or primary producers. So there is a high productivity. And blue carbon storage, when we talk about blue carbon, the seagrasses and mangroves come to the topmost features, and even kelp can absorb carbon dioxide and pass it through the food webs. And the foundation species, as well as the ecosystem engineers, yes, they can change the environmental conditions and provide a very suitable, healthy environment for the other living organisms and shoreline protection. You can, if I said a mangrove, the first thing is you can see a lot of roots, nice roots protecting our shorelines. So, sad scenario is the anthropogenic and climatic impact. So there are like lots of natural disasters can make harm to these biomes. Storms, tsunami, diseases, grazing by herbivores. So these are natural disasters. But the worst case scenario is the human impact or the human disturbance. Other than these listed features or the listed disturbances, the pollution and artificial developments and any type of disturbances done by human can cause a devastating or the degradation of these biomes. So before I go on to talk about this modeling process, my question was what we need to know and where are they exactly and what we can do for the protection. So during this thing, many researchers and scientists have done a lot of work for the understand the biological, morphological, taxonomical, biochemical processes of this marine plants. But we have a limited resources about the distribution, global distribution. So, the next question was how many maps we can rely on, or what are the maps can use for this conservation processes. So there is a map for seagrasses that's accurate, and like I think Frederick Schultz's map available on UNEP WCMC Ocean Data Weaver. So this map is very advanced, accurate, and hard outcome of a hard work. But it's incomplete because this is like real polygons of the patches of seagrasses and the real occurrences. So it takes a long time to complete. And so there are some places need to add more. And so my question is, like, to get a kind of a complete map, can we do something else? And the kelp or the laminarian brown algae, we don't have any existing maps. For mangroves, we do have, it's a satellite image processed map from Giri et al. 2011, and a coral map also developed using satellite images. So all these maps are available on UNEP WCMC Ocean Data Weaver, and all the authors, including myself, describe the limitations of these maps. So what did I do? I collected all the occurrence records and tried to develop a map, a modeled map, to predict the possible distribution around the world, including the countries within limited interest of marine plants, or do they don't have local maps. Also, all these countries covered depend on the other countries' available data. So I use Maxent model and 13 environmental variables available on GMAT, and different open data sources such as GBIF and ORBIS and UNEP WCMC occurrence records. And I cross-validate this with Schultz's polygon layer to verify the prediction of the model. So nearly I developed 1,000 trial maps to get the final map. So, and with this, after finalized, map is look like this. So it covers all the possible or the suitable environmental conditions to occur seagrasses. And the similar and the with experience of the seagrass biome, I developed the same too for the kelp biome to get an idea of the distribution of kelp. So this is version 1. And later on with my supervisor, we developed again a regional map for the Arctic region. And the version 2, we published it. So in the version 2, it has more polygons in the Arctic region. And so the 4th chapter focused about the conservation of marine biomes. So do we pay really a considerable amount of attention to the conservation of marine plants? Or is it enough, or do we need to do more? And where are we exactly? So the first step is to identify the current conservation status of marine biomes, forming species and the IUCN red list. And the next one is the individual area of distribution of seagrass, kelp, Zooxanthellate, coral, and mangrove biomes according to the exclusive economic zones of each country, as well as little biological regions. I divided these biogeographical regions based on IUCN operational regions. So then that is easy to link to where do we need to focus more for the conservation. And then I did analysis to get the multiple biome occurrence within 1 kilometer pixel area, because all these maps I modeled has 1 kilometer resolution. So this is the maximum resolution I think I could go during my PhD time. It's a global level. It's a very, if I could go for 100 meter resolution, this would be more accurate, but it's really time consuming as well as stopped prematurely processing the models. So all these maps used 1 kilometer resolution. And I did the same calculations for the individual ecological EZ zones as well as IUCN operational regions. So this is the outcome of the individual plant species conservation status. So have a look, so you can see the seagrasses are kind of like out of 60 species I used for my study. There is an attention, paid attention to understand the conservation status. So 45 is least concern, but there are endangered species and vulnerable species, and 2 species doesn't have any information. But so the Zooxanthellate corals also like have different conservation status, and almost all of these corals under protect with different conservation agreements and different what do you call it? Some yeah, it's a kind of like agreements from the different organizations and governments. And but the kelp is surprisingly, all the 66 species, only 1 species has record. And I'm talking this 2019, before I submit my PhD. Probably now they updated it later on. But the 66 species doesn't have any information. It's not available on IUCN red list. So there must be some record, at least like data deficient or not, something like that. And so the mangrove species also, they have endangered species, vulnerable species, and the least concerned species a lot. So this is the overall conservation status of individual plants. But now the next step is, can we protect, even it is not individually assessed or not, can we protect it within one large area, or can we demarcate the strict reserves to protect this, including the conservation, despite of the conservation status? And so this is the map of the seagrass biome, the modeled map, and the biome map. And this is the coral biome, coral map, and the mangrove map. So what did I do? I overlapped all these 4 biomes and try to see, is there any places with multiple biomes, or what are the area of individual country has. So when I try to do that, I found it's kind of like interesting when we talk about this Asian Pacific region. So the highest distribution of the seagrass is for the individual exclusive economic zones. The Australia has the highest, following USA and then Indonesia. And the kelp distribution is also the highest is in Australia, and the following USA, UK, and then Japan. So the mangrove is again, it's Indonesia and Australia. And so there's no wonder of the Zooxanthellate corals. So it's Indonesia, Australia, Papua New Guinea, and Philippines. So this is, we can have a clue. So all these plant communities, we have high distribution of all these 4 biomes in Asian Pacific region. And when I try to identify the number of the biomes distributed within this coastal regions, this is the area with 1 biome. So it's like almost all over the world, like it has 1 biome, and at least it's 2 biomes. So this is 1 kilometer resolution, a square kilometer resolution. So this 2 biome areas, this within 1 kilometer, square kilometer, you can see two different types of biomes. And the interesting part is the 3 biomes. So some places you can see 3 different biomes within 1 kilometer. So you can see around this in the Asian Pacific region, there are like lots of red color dots to show these places have multiple biomes. So it's a calculation. So I think the Indonesia has the highest of the multiple biomes, and Philippines, Papua New Guinea. So these are the countries, the topmost 3 biome areas. And so the question is, do we have enough protected areas to conserve, or strict reserves to conserve these the nice multiple habitats? Next one is to see.
3 more minutes, Dinusha. Sorry.
3
more minutes. 3 more minutes.
Okay.
Yeah.
So the next one is I divided it into IUCN operational regions to see what it is and how is the distribution. Now you can see, even though there are like lots of species or the 3 biomes areas, some regions strongly, like we don't have enough IUCN conservation areas. You can see this map shows these are the existing strict reserves, but it's very rare, or there are like lots of limited places. This image shows some strict reserves, like it's not cover the coastal area, but some places like this, it covers nicely the coastal region. Some places like this, it covers. But here, this image shows, it's like there are, this red color shows the 3 biomes, but it doesn't cover the 3 biome areas. So this table shows the percentage cover within our strict reserves. So it's mostly, it's 0, under the 0. So some of this, I think this is because of the into the power 2 square kilometers, so 100 kilometers area. Probably this has more, like at least a 1 or 2 square strict reserves, but it's not sufficient. You can see there's like many of is under 0, so that means we need more marine reserves. So this is my suggestion. At least these countries should have strict reserves to conserve their multiple biomes, and because this giving multiple biomes, giving a protection to multiple biomes, is not only conserving the plants, it's conserving all the other ecological processes, associated faunal communities, and also the juvenile status of the commercial fish species. So it's, if you don't protect this, or if you don't pay your attention to conserve using strict reserves, the harm is not only for the plants, it's for all the living beings of the marine domain. And also the other thing is, it's not just to quantity, the demarcate of marine protected areas is just a quantity. We need a qualitative strict reserves to conserve more species. Thank you.
Thank you. Thank you, Dr. Jayathilake. It's really nice presentation, and then I really enjoy your presentation, working from identifying the biome until the application of this analysis to develop the I really like it, the strict marine biodiversity, strict conservation area, because most of our MPAs, are, although they are called MPAs, but without limited protection. So your research focus on we have to generate or develop the strict protected area. Okay, thank you for that. The next speakers will be the parallel with this with Dr. Dinusha's presentation, and Dr. Qianshuo Zhao will present how the global data set they use for developing MPAs in the world. Okay, Dr. Qianshuo, the time is yours. 20 minutes, 25 minutes. Yep, it is. Yeah. Go ahead, Dr. Qianshuo.
Okay, can you see my slides?
Yeah.
Yes.
Yeah, okay, thank you. Okay, good days, everybody. I'm Qianshuo Zhao from the Ocean University of China. Today, I would like to talk about my recent work about mapping ecosystems and biodiversity-rich spots at a global scale and the West Pacific on a regional scale. So at first, I would like to introduce some background and the motivation behind my research. It was in 2016, the IUCN World Conservation Congress called for the protection for at least 30% of each marine habitat and 30% of the ocean for an effective marine biodiversity conservation. And this goal was again remanded in the CBD's 15th COP conference last year. And so for achieving the goal, the percent is 7.7% this year, and still far away from the goal in 2030. So we would like to know where we could expand the 30% coverage. So in this presentation, I want to talk this topic from a global scale and from a scope of a regional scale in the West Pacific. I did this research in my late PhD period, and published a paper about to prioritize the biodiversity-rich sports on a global scale to include the factors which can indicate different aspects of marine biodiversity as much as possible. I tried to include the ecosystems, the benthic rugosity, which is strongly correlated with species richness and biodiversity, and also the species richness and the biomes, which can serve a strong biodiversity and the realms of species endemicity. And this work was granted paper award. It was granted the Atlas reward by elsewhere for its contribution to the UN's Sustainable Development Goal number 14, marine life life underwater. Yeah. So I will introduce each of the data layers I used in the prioritization analysis. The first group of layers are the ecosystems I mapped in my earlier paper, which is also a global scale work. It was based on an unsupervised cluster analysis of 20 physical, biological, biochemical, and nutrient variables. And some areas belonging to the same ecosystem were geographically divided by continents, but distributed at similar latitudes and symmetrically on both sides of the equator. Each of the ecosystems was represented by an individual presence-absence layer in the prioritization analysis. So that there were 7 layers in total. To give some more technical details, the raw data, the 20 environmental variables were first normalized, and then principal component analysis was conducted to reduce the number of variables and exclude the correlation between the variables, and the k-means clustering analysis using cosine similarity as distance function to do the main analysis to identify the ecosystems and map their distributions. And finally, the mapping was validated by the silhouette values to ensure the proper number of the ecosystems. That is, how many ecosystems should be on the world. The next groups of layers are the biomes, and in this research, the biomes was defined as large geographic areas characterized by the same plant form, which form large areas of 3-dimensional habitats with high primary productivity. And thanks to Dinusha, I can get the global distribution of seagrass and kelp, and I also collected the distribution of mangrove and the shallow water coral reefs from the same data set. Specifically, the shallow water coral reefs was recognized as biomes because they can host photosynthetic algae and provide a complex habitat structure. The layer of rugosity is derived from the depth data. It was based on the depth variation of the cells around the targeted cells. So the higher the benthic roughness, and normally the higher the biodiversity condition. And the layer of within realm species richness is a little complicated. It was synthesized from species richness and species endemicity realms. The purpose of the synthesis is to know the condition of species richness within realm, within each realm, through comparing the species richness numbers within the same realm rather than the ones from the other realms. By including realms, we can recognize regions of greatest difference in species composition. Richness, the species richness alone does not consider which species are present. After collecting the data layers, they were introduced into the software zonation for the prioritization analysis. The basic algorithm of zonation is to iteratively remove cells from the lowest significant cells up to the highest ones. In this research, the removal rule is the target-based function. This method prioritizes the cell which can contain the presence from multiple layers rather than a cell which only cover an extremely high cell value from a single layer. Specifically, because to satisfy the goal of this research, I set a target of 30% of each layer and the general analysis, which means the highly prioritized areas are expected to cover 30% of each layer. So this is the primary result of the prioritization analysis. From the light red to the dark red, they represent the biodiversity from the lowest to the highest in the 30% most highly prioritized areas. The prioritized areas were mainly located at the continental coasts, island arcs, oceanic islands, the Southwest Indian Ridge, the Northern Mid-Atlantic Ridge, the Coral Triangle, Caribbean Sea, and the Arctic Archipelago. This post-analysis is to test how well the prioritized areas covered the layers used. The prioritized areas generally achieve 30% coverage on presence-absence layers and fairly covered the top percent of globally covered layers. These are the figures of the comparisons with current marine regions. Only few prioritized areas were covered by current MPAs. For the no-take ones, the coverage was even less, 1%. Therefore, more MPAs could be proposed based on the prioritized areas in A, B, NJs, and EEZs. So this is further work conducted by Tamlin and trying to explore if we want to protect 30% of the range of each threatened species, how much area should be protected worldwide, and the result showed at least 40% of the ocean should be protected for an effective protection for the threatened species. In addition to the presented work, there are in total 5 data-driven marine prioritizations, and if we want to do some immediate protection activity on global scale, we could directly to plan and establish MPAs in the consensus sites, and there should be no doubt, I think. And in the second part, I would like to further talk about the work in the West Pacific. Following the similar approach, I mapped the surface ecosystem and the benthic ecosystems in the West Pacific region. The study area in the West Pacific region was delineated by the EEZs of each country and their regions here, including the high seas in Philippine Seas and the Tasman Seas. Yeah, and based on the ecosystem mapping, we further explored their relations with distribution, with species distribution, and some further relations between the environments and species community, species composition, and richness here. This slide is an example. We explored the relation between the temperature and the species richness, the area size of each ecosystem, and the richness in each ecosystem. And we can find in the West Pacific, the temperature is generally positively correlated with species richness, similar to the condition on terrestrial ecosystems. And but for the comparison between area size and richness, the correlation is not obvious, or there is a slight negative correlation between the size and the richness, not like the condition in terrestrial ecosystems. And for now, we assume it may be because in the ocean, the marine species can move more widely than terrestrial species, and they have a wider distribution. This may be the reason. And similar to the global work, we also collected the distribution of the 4 biomes, species richness, and benthic ruggedity in the West Pacific region. And as Dinusha mentioned, we can find the data of the 4 biomes in the UNEP WCMC Ocean Data Viewer from this data set. So this is an initial analysis outcome of the biodiversity prioritization in the West Pacific. So in this prioritization analysis, it covered the factors of ecosystems, species richness, the benthic ruggedity, and species endemicity and biomes. And a similar post-analysis was also conducted, and this this analysis also will cover 30% of each present absence layers and fairly achieve the goal of covering 30% of the continuous layers. This is a brief comparison between the prioritized areas in the West Pacific and the current MPAs, and we can see that for the no-take MPAs, there is only a very small overlap. So which means we may have to do some more work for an effective protection. And in addition to the West Pacific, we are also trying to do some to do the regional scale work in other places, such as the Marine seas in Europe, and I and my team members are collaborating with Mark Costello to do a similar project in the European marginal seas, adding some local features and blue carbon factors, something like that. And we are trying to do, want to see whether there could be anything different in European seas. Okay, thank you.
Thank you, Dr. Zhao. It's really nice to see your work because previously my work is only in the Coral Triangle region, and now we have a maps for whole world. Yeah, and then it is really great that there is a I know that there is a 30 by 30 movement that we need to protect 30% of our ocean in the 2030, and now you have a Dr. Zhao provide us with very clear and very solution where to protect, which are that need to protect if you like to achieve that 30%. Thank you, Dr. Zhao, for your presentation. Okay. It is now we are moving to the next steps of our seminar today. Is question and answer. I know, Dr. Zhao, if you would like to answer one question in the chat box, maybe you can answer it live. There is a question from Anthony Carlos that I will read it to you. Maybe you can respond. Maybe you can respond the answer first. Before we give the floor to other participants.
Yeah, I'm reading the question.
Yeah.
Okay. Thanks for your question, Antonio. And the approach I use for the prioritization analysis is it is an open method, and in addition to the consideration of biodiversity conservation, you can add the other considerations in your specific project, such as you want to consider social and economic aspects. You can quantify them as data layers, and you can introduce them into the analysis, such as the cost layer or a normal layer like the other data layers for prioritization. So I think that depends on what you really need in your project. And so as far as I know, this this algorithm, this software can contain much more layers if you want. So I think that is not a problem, and once you can assure what you want from the local stakeholders, so you can introduce their consideration in the analysis. Yeah, thank you.
Thank you. Thank you, Dr. Zhao, for your answer. Okay, if there is any question from the participants, you may raise question now. We are in the question and answer session. There is a question from Takeshi Yamakita.
Yeah, actually from me.
Yeah.
So the question is about the data bias, because all of the data has biases, and I think Jasia also mentioned it will be updated recently. So if there is biases, did you use any indicator that mitigate it, or do you have suggestion to facilitate more to obtain more information or eliminate some taxa which are biases? For the both of you.
Thank you, Takeshita. Dr. Jayathilake, you want respond to that question on bias of the data and how to mitigate the bias?
Yes, so this one is quite interesting, because when I worked on the seagrass, especially the seagrass is showing a regional distribution, I should say like that. So the specific species live in a specific region. So this was a challenge for me to avoid this data bias. And the other thing is, like, there are lots of occurrence records come from the developed or the temperate region than this in the Pacific region, especially the Indian Ocean or the South Asian region. We don't have much records. So the problem was whether my model is representing the real distribution or it biased towards the temperate region. So what I did to, it's very difficult to do a kind of like a normalization or this, because we have I'm doing global level and every single record is important. So what did I do? I tried to develop different types of models to see, is there a really data bias account in this model, to see is there an impact of this data bias. So I selected the same number of or to the latitudinal same number of occurrences for each latitude and try to do a model, but it didn't predict the distribution. So the real prediction, or the final map was developed using all the occurrence records. I think because we have nearly 40,000 occurrence records and the range is wider. So the environmental range is wider, and this calculates the highest probability of occurrence. So it in my map, I didn't see any of this, the impact or any side effect of the data bias, but I highly recommend to not to go for the because for the global conservation or to get a picture of global distribution, I developed this map. But in my research paper, also I recommended it is better to do a local maps to the local models and then combine it. Then we can avoid this data bias or anything, any kind of impact of the different environments, or the impact from the different environments, or the availability of records or something like that. But that was not happen to my kelp biome, because the kelps are temperate and we have enough records for the so the kelp biome is it's temperate, and we have enough records for the temperate region. So that's how I handled. And the next question is like where we can give more occurrence records or something like that. So I think when anyone can upload to GBIF or Orbis. There's a routine of or the process of submitting new occurrence records. And I think you know more than that than myself, because I downloaded occurrence records from GBIF and Orbis, but I think you know how to upload occurrence records to that. And are there any criteria to selection resolution just beyond the limitation of calculation time? Yes, so I think the resolution time is depend on resolution, or the resolution of a map depend on the objectives of your, is that the what? Or am I going to the next one?
We can finish the first one first, Dinusha.
Yeah. So the more data in the future, or where are some taxa more biased than others?
Which taxa that you need to add more data?
Yeah.
Do you have any suggestion for that one?
Sorry?
Do you have any suggestion that which taxa that need to be more data?
Yes. So some seagrass species. Overall, any species in Indo-Malayan or around South Asia, Indonesia. So this region, if we could get more occurrences, it's better, because if you see the occurrence records of these seagrasses, Indian Ocean is the least. In the West African region, we don't get enough occurrences. So probably there are, but we don't get so sufficient amount. So this is the aim of my work, to fulfill that gap from the outside occurrences, and to draw polygons to see the possibilities of distribution. Yep.
Thank you. Thank you.
Qianshuo, do you have anything to add?
Qianshuo, do you have any? Or Qianshuo, can you move to the next question on the spatial grid? There's also a question on resolution of your spatial grids then affect the analysis, because I know that you are more get into this spatial grid. We have a long discussion this kind. Okay, can you explore more that one in regard?
Okay, thank you. In my experience, so the difficulty of researching on a global scale, so not much data can be used on a global scale. So such as Dinusha mentioned, in some sea areas surrounded by developed countries, so you can have adequate data, but in somewhere else, it would be a challenge for global scale data. So in such a situation, usually the environmental data can be more easily collected from a remote sensing data set. It is not a problem at present, I think, but the difficulty is the how to collect the biological data. So I think for the proper resolution, it is primarily depend on the environmental data you collected, I think. To be honest, for myself, I did not meet a question of the calculation time, because on a global scale, because the true difficulty for me is the insufficient of available data. Yeah, this is on a global scale research, and I think, yeah, on a regional scale or a local scale, we may choose a proper resolution to balance the accuracy and calculation time, I think. Yeah, and I see a new question about how to contribute more data at local scale. I think you may have heard the data set OBIS and GBIF. So they are open sources, and any data contributor are after a basic eligibility assessment, I think they can contribute their own data to the open source data set if they want, and the data they contributed can be used for a local scale research, and can also be used for a regional or a global scale research that depends on the users. So if you have, if we want to contribute, my suggestion is you can try the OBIS dataset and the GBIF dataset. Yeah.
Thank you, Dr. Zhao, for your clarification on the data sources. Is there any more questions to these two panelists?
One person asked about resolution of the
the limitations
of
resolution.
My idea is for marine plants, it is better if we could go for 100 meter resolution, so that will avoid the blank spaces coming to like 1 kilometer resolution. 1 kilometer resolution is like, it is a big area, and some places you can get this 1 kilometer distribution, but many places you don't get it, but it's added to your distribution area. So 100 meter resolution is enough for the marine plants. You can get a kind of like approximate or more closer to the real value. But if you, it depends on what species you are working. So if you are going to, and the distribution range of the species. And for example, like mapping huge forest areas, you don't want to get a 100 meter resolution. You can get, when we talk about the terrestrial forests, probably like 1 kilometer is accurate. But because the difference for the 1 kilometer is like we can consider, but for marine, 100 meter is better if you could go. And it depends on the range of the distribution of the specific species. If you're going to work like a snail species in this little stream, there must be a very, very finer resolution. And if you're going to work on the weather or something like that, that will be like a big or the very coarse resolution. Yes. So this is my answer.
Oh, yeah. Thank you, Dr. Jayathilake, for your
to add information. So in my own experience, if your research is on a global scale and is associated with biological factors and environmental factors at the same time, I think a resolution of 0.5 degree is sufficient. Is sufficient very much, I think. My own experience.
Yep.
Agree with you. 2 more questions on that.
Yes,
I
there
think
are some.
There are. I think we already answered. Dr. Qianshuo already answered at local data set, and then there's one from Carlos. And the restoration project. Yeah, is there any possibility to downscale your data, your biome data that we can fit to the local restoration purposes?
Oh, I think because this is a spatial distribution map, so it's very hard to use the temporal changes. And the only thing is like the modern maps depend on the existing point records. And if there is a change for the future versions, that means we need more points, and then that will be more accurate. But the best thing is to monitor restoration project is to use the satellite images and do a temporal change. I think that is the solution other than our biomaps. Our biomap has the limitation to like, it's not a temporal updated ones. Even like we try to do it today, we have to depend on the existing records, but that is not a, it doesn't give any significant change to the model map. The only thing is you can use for seagrasses, there is a latest update of the Frederick Short's map. It's updated 2021. You can use it. It's on the UNEP WCMC Ocean Data Weaver. You can use it, but it's also with the submitted records. So yes, the best thing is you need to use satellite images and do an NDVI to get the change.
Cool. Thank you.
That's my opinion.
Yeah,
that's what I
really
will
cool
do for that.
Yeah.
And I'm involved.
Thank you.
Yeah.
Yeah.
I don't
Thank
know
you,
the
Dr.
other one.
Jayathilake, for your explanation. Yeah. There's one question for Dr. Zhao.
Yeah, I'm reading.
Yeah.
Yeah.
Okay, it's about temperature. Seawater temperature.
Yeah, I see. Yeah, the current research cover a large geographical range from the very north to the very south across all the West Pacific. So it do cover the temperate and trophic areas, and this is the current result we found, and we are exploring the rule or logic behind the analysis outcome, and yeah, I'm not really sure whether it related to the recent warming, and yes, we are also trying to collect predicted environmental variables in the West Pacific, and to see would there be any interesting variation under different climate change scenarios, and yeah, and how the ecosystems would vary in their environmental variables values or the associated species distribution. Yeah, we are still exploring.
Thank you, Dr. Zhao. Yeah, all these things is there any more questions?
Yes, I'm answering one question for the
Yeah,
like,
you're writing the answer.
yeah.
Yep.
I think it's the same one. I already answered.
Yeah, it's the same one.
Well,
Yeah, you already responded to that one on the restoration project, Dr. Jayathilake. So, yeah, participants and the panelists, we have seen that the opportunity to use the point occurrence data record to develop a model, partial distribution model, to point out where is the location for the developing the MPAs. And then there are a number of points that we discussed today. From the technical part, it's very nice discussion on which resolution that we can use, from local, regional to global, and what kind of data set that we can use to develop that one. That we have to realize that there is an underrepresentation of our data sets in global data sets. There is a spatial scale underrepresentation. There is a record-based underrepresentation. There is a temporal scale of underrepresentation that we have to, that's the best data that we have that available now, then we can use that one. Dr. Jayathilake and Dr. Qianshuo Zhao shows how shows a result of their project, of their research, that we can use that data set to develop a really nice result, a really nice formulation that, this is the area that we can develop our MPAs or marine protected areas. I think that's it's not a conclusion for our discussion today. See you on next Friday, and get back to the host. Thank you very much for your attention, and I wish you all healthy and luck. Thank you, Dr. Zhao. Thank you, Dr. Dinusha.
Thank
It's
you.
really nice to meet
Thank
you.
you.
Thank you very much.
Take Yamashita, Masa Nakwako, and Juan Carlos. Thank you very much.